Welcome back to our 5-minute dive series of Proteins Unfolded, the podcast where we explore the extraordinary world of proteins and how they shape life as we know it. I'm Andrea.
And I'm Emilio.
And we're researchers at the Human Protein Atlas based at SciLifeLab Stockholm. In today's episode, we are tackling another fascinating question. What is a secretome?
And what does it tell us?
One way to start answering this is thinking about the body as a busy city. In any city, there's activity inside the buildings, offices, factories, homes. But there's also a lot happening between the buildings, deliveries, roadwork, communication lines. The secretome would be everything that leaves the buildings and enters the city's shared space.
More specifically, the secretome refers to all the proteins that are actively released or secreted by cells into their surrounding environment. These proteins aren't just leaving the cell for no reason, they're meant to interact with other cells and tissues. In fact, they can even travel through the bloodstream and act in distant parts of the body.
Exactly. A great example of this are hormones, which carry messages between organs. Just to give you one example, the corticotrophin-releasing hormone, or CRH, is secreted by the hypothalamus, a region of your brain. CRH is an interesting hormone because it's basically a stress driver. When CRH is released, it leads to the release of the adrenocorticotropic hormone, you can say that again, from the pituitary gland at the base of your brain.
These might still be seen as close interactions. However, the adrenocorticotropic hormone actually travels in the bloodstream all the way to your adrenal glands on your kidneys, where it causes the secretion of cortisol, aka the stress hormone. So, as you see, the secretome is no simple matter. It is responsible for a lot of basic processes. Even the enzymes that break down food in your stomach are part of the secretome. Without secreting molecules, cells couldn't communicate or coordinate their activities, and the body would fall apart.
Now, if the secretome is everything leaving the cell, then we should also talk about what stays inside and what stays at the door. This is where the intracellular proteome and the membrane proteome come in. The membrane proteome refers to proteins that are ingrained in the cell's outer layer or membrane, while the intracellular proteome are proteins which stay within the cell. Both of these proteomes have their own functions. For example, membrane proteins can act as gatekeepers, controlling what gets in or out and sending signals between the inside of the cell and the outside world.
So you can actually think of the secretome and the intracellular and membrane proteomes as three parts of a conversation. The secretome is sent out to communicate or influence its surroundings, while membrane proteins receive incoming messages and pass them along using the intracellular proteome.
However, if they're all important for cell communication, why do scientists separate the secretome when studying proteins? With us today, we have Åsa Sivertsson, a principal investigator at the Human Protein Atlas in charge of the Structure and Interaction section. Åsa, why do scientists study the secretome and the membrane proteome separately?
That's a good question. The answer lies in function and context. The secretome is especially important for understanding processes that involve large-scale coordination across tissues or organs, things like immune responses, wound healing, or even cancer metastasis. Meanwhile, the membrane proteome is all about the first line of interaction. These proteins are the faces of the cell, the ones involved in immediate sensing and signaling, or in allowing nutrients in and waste out. One practical reason for separating them is that the secretome is easier to study in body fluids like blood, while membrane proteins are embedded in a fatty layer, making them trickier to extract and analyze. And intracellular proteins require us to break the cell. Scientists often focus on one or the other depending on the research questions, and the insights we gain from studying each are different. For example, mapping the secretome can help us identify biomarkers in the blood, proteins that act as warning signs of disease. The membrane protein, on the other hand, is crucial for drug discovery because many medicines are designed to target membrane proteins directly. So if you imagine a scenario like cancer, studying the secretome might help detect the disease early or track how it's progressing, while studying the membrane proteome could reveal potential drug targets to stop the cancer from spreading. This is why the secretome and membrane proteome are often studied side by side. Together, they give us a fuller picture of how cells interact with each other and their environment. The more we understand these systems, the better we can design new diagnostics and therapies.
Thank you so much, Åsa. If you're curious to explore specific proteins in the secretome, check out the Human Protein Atlas website, where you'll find tons of fascinating data on the proteins that make communication and coordination in the body possible.
So that's it for today's dive into Proteins Unfolded. If you enjoyed this episode, be sure to share it with somebody who loves a good science story.
And if you have a question you'd like us to discuss, send us a message.
See you next time.